So we had a lot of great sort of offline discussion about like theta and what does it mean. So I'm going to go into a little bit more detail here into this idea of the theta oscillation serving some essential computational function in the hippocampus. And so the first thing I'm going to do is going to point to this. It's a simple model that illustrates a property, which I didn't show here. And so here, a couple of things that you see. One, you see the phase locking of hippocampal spiking theta oscillation. You see this long-range coupling, that is coupling phase locking of prefrontal cells to hippocampal theta, suggesting that theta serves as a long-range coordinating function with other areas. But third, you see a property that John O'Keefe identified, again, in the early 1990s. I don't know if they actually mentioned this when he got the Nobel Prize, but it's a property that I regard as equally or perhaps more significant in understanding hippocampal coding than the spatial firing, the place fields. And what O'Keefe noted is that, again, when the animal moves through its place field, it produces spikes. The spikes are locked to the theta oscillation. But as an animal enters a field, there's a particular phase at which spikes seem to be expressed. When the animal first enters the field, firing is low. Again, remember, think this long tail as you kind of come into it. Cells, they start to fire at relatively low rate, and then the firing rate increases. Well, that's what you see here, this kind of asymmetry. Start low, few spikes, and then you get to a higher firing rate. That's the idea. But what O'Keefe noted is if you look at the timing or the phase of the first spike of these little episodes, here, the first spike here as the animal enters the field, here fires at the peak, and then the farther into the field the animal goes, the earlier and the earlier the first spike occurs. And that is that there is a relationship not only between firing rate and position, but firing phase, or theta phase and position. That if you know the phase of firing, then you know the relative distance within the field. Late phase means you're just entering the field. Early phase means you're sort of at the peak of the field. And he termed this property phase precession. Now, interestingly here, I was just noting when we were talking about this offline, if you look at these prefrontal cells, you see this prefrontal cell shows the same property. This spike here fires at a late phase, and then each successive cycle of this theta oscillation, the cell seems to fire at an earlier and earlier phase, suggesting that the prefrontal cortex also expresses this property of what O'Keefe termed phase precession, and as a change in preferred phase over time. In this case, change over time corresponds to a change in position because the animal is moving. And so if we look at this, so this is some actual data. If you do this repeatedly, the animal goes through that place field a dozen times, and you just kind of accumulate all the spikes as a function of position, but then you also measure the firing of those spikes as a function of theta phase. This is the phenomenon described as phase precession by O'Keefe. And that is, as an animal is entering the field, so an animal running from left to right, just for the zoomers, I'll put it on here, animal going from left to right, again, repeatedly. And then you're asking, when do the spikes get emitted? And what you find is, okay, you see here spikes at relatively low rate, and the number of spikes is relatively low, but their timing is such that they're always occurring late in the theta phase. And then as the animal moves through the field, going from left to right, spikes fire earlier and earlier. This is phase precession, the advancement of phase as a function of position. But you also notice there are a couple of other things that are going on here. So not only is phase changing, but also, again, the rate, spikes over here. There are far fewer spikes over here than there are over here. Far fewer spikes over here than there are over here. And also, if you now just if you sort of mentally, let's just like split this in half. Just focus on, ignore the lower half, that is from 0 to 180, and then just look at the organization of firing, of spiking in this late phase, 180 to 360. And what you see here is that there seems to be a strong linear relationship, strong correlation of phase and position. This was this property of spatial coding that O'Keefe initially sort of attributed to phase precession. In fact, he described that when he found this, he sort of revised his evaluation of spatial coding in the hippocampus to say that space is really coded in phase, not in firing rate, and that firing rate actually captures velocity. This is the way he described it. So his view of hippocampal coding was that the real spatial information is in the phase, because he saw this strong linear relationship. So the thing about the firing rate is you say, well, if you know that the cell has fired, you can tell that it's here, that's somewhere in this field, right? That's what you can tell. But if you know exactly when the thing fired, now you know with very high temporal precision where it is in the field. But that really only applies for this part of it, 180 to 360. So now just, mentally forget that, block this out, and now look at the 0 to 180 part. Okay, what do you see there? I don't see a strong linear correlation here, right? It just looks like there's a lot of spiking, but there's very little phase by position information. It's just, right? It's just all distributed over here. So you can really think of this theta phase coding as having these two domains. This late phase domain in which you have low firing rate and high temporal precision, what I refer as the temporal coding domain. And then you have this early phase domain in which you have high firing rate, lots of spikes, but very little temporal information. And this is kind of the trade-off. You say, look, if I want to get timing information from spikes, then I can't have too many spikes, right? If I have too many spikes, because producing spikes takes time. And so the only way I get timing information is if I fire at low rate. So here I have low rate, high temporal precision, late phase, high firing rate, low temporal precision, early phase. So you think of there being sort of two coding domains within the theta oscillation. And so you can ask, okay, you can kind of describe it that way, but how would that mechanistically, how might that actually come about? And so here's a simple model that argues that these properties of phase coding can arise from the simple biophysics of individual cells. So you can think of there being two processes that govern the firing of spikes. One, excitatory drive, shown here, for instance, by this blue line. There are actually two cells here. I think I only wanted to show one, but just think of, first focus on this one, the blue cell. So you have excitatory input that is changing as a function of position. So you imagine there's a hippocampal place cell, it's getting spatial information, spatial input. As the animal moves, that spatial input changes. And we're now just going to sort of describe that change here as this kind of this spatially asymmetric or ramping input. And then in red, you see a complementary process, which is time varying here in red, and reflects the relative excitability governed by inhibition. So you've got excitation changing as a function of position, and then you've got inhibition changing largely as a function of time. And the simple biophysical principle you're going to apply is the idea that an action potential is going to occur when excitation exceeds inhibition. So when the blue line is higher than the red line, you can get a spike. If red line is higher, inhibition is higher, you don't get any spiking. And so you can ask, if you have the spatially asymmetric input along with time-varying inhibition, when are spikes going to be emitted? And what you see is that, well, with this model, the first spikes, and that is with weak input, will occur at late phase. You have to wait till inhibition drops all the way down to here before you emit a spike. As the excitation gets stronger and stronger, that point becomes earlier and earlier, and that is now excitation exceeds inhibition here. And so you can both fire earlier and you can fire more. And that is the blue will be over the red for a longer period of time. And so here, earlier firing, fewer spikes because of the shorter time window. Here, or late firing, fewer spikes. Earlier firing, more spikes. Simple biophysical model. Now, I think that the sort of the power of this encoding principle only becomes clear when you add in a second cell. So now we have a second place cell, apply the same principle. It's just spatially, the input is now spatially offset. So this has got like a ramping input, it's just now over in this location. And now you ask, when does the blue cell fire and when does the purple cell fire? And what you see here is because the input has been spatially offset, blue cell will always fire before the purple cell. Place cell 1 will always fire before place cell 2 on every theta cycle. Every theta cycle. And so what you actually have is so phase precession for single cells translates into theta sequences across cells. And so if you elaborate this now, instead of having 2 cells, as I mentioned, at any given location, you typically have a few thousand cells. So now you imagine what you have here, every theta cycle, you have a few thousand cells that are going to kind of populate this sort of phase range using this principle. And so if you look at actual data, so you say, okay, we record a bunch of cells. Is that actually what you see? This was some stuff by Dave Foster. It's just like raw data. So completely unprocessed. This is just a single pass, animal going from left to right on one of these linear tracks. And so what you see is this is the spiking activity across, maybe a dozen cells or more. And what you see are these little windows where you see theta cycle scale windows, where you see these little sweeps of spiking, suggesting, yeah, multiple cells on individual theta cycles seem to be distributed across theta phase in a way that's suggestive of a sequence. Now, if I just showed you that, you might think, I don't know, is that really a spatial sequence? Well, to argue that it's really a spatial sequence, you can apply this Bayesian decoding. So you say, okay, I'm not going to show you the spikes in their order. What I'm going to do is I'm going to take the spiking, I'm going to apply this kind of Bayesian, right, the inverted conditional probability to estimate the probability, now using a much finer time window, 10 milliseconds in this case. And so now I'm going to decode estimated spatial position as a function of spiking at 10 millisecond resolution. Of course, the theta cycle is about 100 milliseconds, so now I have sub-theta cycle resolution. And when you now do this decoding, so the gray here now indicates it's the estimated probability of being in a given location as a function of the population activity at that time. And what you see here, so this, I would argue, is the code. This is the spatial code in the hippocampus using these decoding methods. And what you see here is that you see, this is just the raw data, sort of a single second or so of theta modulated activity. You decode at 10 milliseconds, and what you see is that the spatial code now sweeps from a relative position. The dotted line shows the actual position of the animal. The grayscale shows the decoded probability of being at a position relative to the animal. So the hippocampal goes from just behind the animal to just in front of the animal. So 10 times a second, what you have is the hippocampal code is sweeping from just behind the animal, in this case, a position, this is like, what, 60 centimeters or so, just behind the animal to just in front of the animal. And if you now average across all the theta cycles, so I take all these theta cycles, I average the estimated position as a function of phase, I get this. So this diagram simply shows what is the relative position decoded as a function of theta phase, and this just kind of illustrates this. So it goes from just behind the animal to just in front of the animal. But there's a couple of other things about this. One is you see it's not like strictly linear. Again, there's a sort of high density region. There's kind of it suggests that that there's a difference in the relative position by phase. So this is like phase, change in relative position. Here's change in phase, change in relative decoded position. So high fidelity, low fidelity. And so kind of thinking about this, so this suggests, yes, there's coding of relative position as a function of phase. There's sort of a differential coding of sort of relative position of relative predicted positions as a function of phase, and that's sort of the correlation of phase with position as a function of absolute phase. And so you can, using this using this illustration, you can think of there being like two, this is like the early late phase thing, early late phase. But now you're thinking about it not in terms of the cartoon, but in terms of the actual decoded information. And again, what I'll show you is that you can kind of split this into two functional domains, what I'll describe as the temporal coding and the rate coding domain, and then demonstrate that these different domains have different behavioral consequences, that you can manipulate activity in these two domains and get different behavioral outcomes. So I'm just going to kind of quickly run through this. I already showed you, this is these recordings from the hippocampus and prefrontal cortex. And just kind of functionally, both the hippocampus and the prefrontal cortex I mentioned, they're both at the top of the processing hierarchy. So like complex behavior, complex decision-making, navigation being one of those, they're both affected by damage to hippocampus and or prefrontal cortex. It's not surprising. You need both. You need, you need to kind of construct complex models of space, but then you also need to use those models to direct complex behavior, that is direct navigation in the prefrontal cortex. And a simple version of this kind of spatial decision-making, of a spatial working memory or spatial decision-making task is shown here. This is like a T maze where you can direct the animal to make a choice at this at the T junction, and that can be based on different things. You could base it on a cue. In this case, we're basing it on sort of the previous behavior. Alternation is one configuration. That is, if you went left last time, then go right the next time. So you have to remember where you've been in order to remember where you're supposed to go next. Very simple. And so we're just going to record activity in the prefrontal cortex, in the hippocampus, in those, well, this in a task like that. This just kind of illustrates the activity that's been associated with the hippocampus, the prefrontal cortex, in different sorts of tasks. One of the properties of prefrontal activity, one of the functions of prefrontal cortex that's often described as this property of working memory, holding information in mind in order to make a delayed decision. Again, related to this task that I described, spatial working memory, where you have to hold in mind where was I last time in order to make a decision about where do I go next. And so just kind of recording from these areas, I already showed you this, just to illustrate that you have firing in the prefrontal cortex that seems to relate to the hippocampus through theta phase locking. So it seems like the prefrontal cortex might be listening to the hippocampus, and the theta phase might be relevant to that. And you can analyze this. A variant of this task, of that T-maze task, is shown here on the left. This is the variant is, this is a back-to-back T-maze task. So it's two T-mazes back-to-back, and the variation is that there are two directions to this. The animal's going to start at one location. It's going to start at like on the left side. It'll start up here, kind of run down the center, and then we'll have to go either left or right. So the animal starts on the left and then runs to the right and has to choose. And then once it does that, it'll turn around and it'll go back. But when it goes back, when it gets to this point, it doesn't choose. We just force it into one of these arms. So it's a continuous spatial working memory task in which the left side is like the sample. That's where you start. And the right side is where you choose. And what's interesting about this is that it's behaviorally symmetric. The animal runs, it goes to the left or the right. Here it runs, it goes to the left or right, right? It starts in one arm, makes a turn, goes left or right. It's just that in this direction, the animal chooses, and in this direction, the experimenter chooses. And so the question is there any difference? What goes on here? What's the difference in hippocampal prefrontal interactions when the animal has to choose or when the experimenter has to choose? And so this is the idea here. Choice, we choose, the animal chooses. And you can look at a couple of properties. One that I sort of showed you. One is the relationship of spike activity to the local field potential or the theta oscillation. So here you have the hippocampal local field potential, the theta oscillation, and you have spiking in the hippocampus and spiking in the prefrontal cortex. That's what we're going to be looking at. And so we can ask, so in this task, when you're looking at like spiking activity in the prefrontal cortex relative to hippocampal theta, and that is phase locking of prefrontal spiking to hippocampal theta. When does that occur? And there are three conditions. One, again, we're looking in that central arm. In gray, you see this is the degree of phase locking of prefrontal spiking when we choose. The animal's making no choice, it's running on the track, it's gonna go left or right, but it doesn't make any decision. In red, in solid red, you see this is the degree of phase locking that you get when the animal's going in the choice direction, and it makes the correct choice. And that is that it went the right, it remembers where it started, and it makes the correct choice. Interestingly, here in red, but in the hatched red, this is the degree of phase locking when the animal's moving in the choice direction, but it gets it wrong. So what this suggests is that it's not just the two directions, it's the accuracy of the choice, and that is, did the hippocampus, did the prefrontal cortex, did it listen to the hippocampus? Was it able to communicate with the hippocampus, therefore solve the spatial working memory task? And that condition is indicated by the presence of this theta phase locking. And that's to say that suppose I'm just recording in the brain. I'm not watching the animal. I don't know what the animal's doing. All I see is, like, I got prefrontal spiking, I got hippocampal theta oscillation. And as I'm watching, I'm saying, hey, look, I see good phase locking. Looks like the animal's on track, is gonna get this right. On the other hand, I see crappy phase locking. I say, the animal is not gonna get this right? It's not gonna make the correct decision. I could predict the animal's choice accuracy by simply looking at the degree of prefrontal hippocampal theta phase locking. Now, this doesn't appear in the hippocampus. The hippocampus just is phase locked in all the conditions, which is, as I was saying, the hippocampus is not the CEO, it's not the executive, it's not making the decisions. The hippocampus doesn't know whether you got it right or wrong. It's the same, looking at hippocampal phase locking gives me no information about choice. Looking at prefrontal hippocampal phase locking does. Yes, so I don't show the data on here, but actually what you see is, I'll show it here. Interestingly, where you see it transiently appear right here, and this is in about the 500 milliseconds just before it actually reaches the choice point. And as soon as it makes the choice, if you actually look at that degree of phase locking, for instance, here in these regions, it's gone. So it's only present and only predictive here in the area prior to choice. You don't actually see it. Once the animal starts to make the turn, then that phase locking goes away. That doesn't mean that it's not meaningful. It's like, it just says, if the phase locking was indicative of some kind of transient communication with the hippocampus that was used to guide decision making, once the decision has been made, the prefrontal cortex doesn't need to listen to the hippocampus anymore. That phase locking drops away. So it's transient and is limited to the region just prior to that, the choice point. And then interestingly, again, it's this, if you look at the timing, it's not the whole thing, it's really about 500 milliseconds before, which is, it's like 5 theta cycles. That's a great question. I think the question is why just lock here? I think the argument that I'll make is when you actually look at the nature of what's the activity look like when it's phase locked here, let's say 500 milliseconds before. And what you see is at this point is where you're going to be getting sweeps. This is where you're going to get these predictive sweeps. And so the predictive sweeps at this point are just going to be able to make it to the choice point. And so the idea is you start to lock at the point where the hippocampus can actually provide you with useful information about the trajectories. At this point, that information might be limited, but as I didn't actually show it here, but I described it. In a task like this, when you look at place cells at points like this, they also split. They're not just, when I say, you get directional firing, a cell that will fire when the animal's in this location going this direction versus this direction, but you also get cells that are trajectory dependent. A cell that will fire when the animal's here going in this direction, and it will go left and it will go right. So the left and right trajectories in the hippocampus bifurcate at this point. You get different hippocampal representations for the two. And so the idea would be the prefrontal cortex kind of locks to the hippocampus at the point at which you get this bifurcation of hippocampal trajectories so that okay, there's the left, you can think of it as like the context, left context, right context. which is the right one, and then so the prefrontal cortex evaluates and then decides based on that. And so you can imagine, yeah, if you could like extend that, you know, the bifurcation in the hippocampal representation, that's presumed that would determine when you would actually get the phase locking. It's not a fixed time, it's really based on the sort of the bifurcation of the hippocampal representations. Yeah. So you have phase locking, the spike to phase locking, which is predictive of performance, but you can also look at the local field potential. Now, as I mentioned, you don't necessarily have to have the local field potential, but when you do get field potential in the prefrontal cortex and the hippocampus, you can ask, is there coherence, and is that coherence predictive of performance, and is the coherence restricted to any particular frequencies? And what you see here is that if you look at LFP to LFP coherence, choice versus forced in gray, choice in red, sort of the same breakdown. There's the forced direction, the choice direction when you get it right, choice direction when you get it wrong. And what you see is that you get the only significant change in coherence is for the choice correct direction in the theta band. So the idea that theta coherence is the condition that is both sufficient and necessary for indicating, you know, effective hippocampal prefrontal communication that guides choice. Now, when I say, you know, sufficient, necessary, you say, geez, man, that's kind of strong, right? I mean, you didn't really show that it's necessary. You didn't make any sort of manipulation. So ideally, what you'd like to do is you'd like to go in and test this. You say, okay, you need to manipulate activity as a function of phase and show that phase really matters. This just shows phase is correlated in some way with performance. But can changing activity as a function of phase change behavior? And so that's where these optogenetic manipulations come in. So Josh Siegel, when he was a graduate student in the lab, kind of applied methods that allow you to disrupt activity in the hippocampus as a function of theta phase. So it's a closed loop manipulation. You're measuring theta phase, and then you're optogenetically activating inhibitory cells, shown here in like blue. So you can transiently activate inhibitory cells, and that shuts down pyramidal cells. So what this means is you can kind of shut off, you can transiently lesion effectively in a reversible way at the hippocampus. Turn off hippocampal output for limited periods of time, and you can limit those periods of time to certain phases of the theta oscillation. And when you do that using exactly the same task that I just showed you before, same task, it's the same idea. We're going to look in this central segment, choice direction, force direction. Now, the thinking was, okay, we're going to test this whole working memory idea, right? That you actually require, that the prefrontal cortex requires phase-locked hippocampal information in this region in order to make the right choice, right? So if you shut it off, you turn off the hippocampus here, where the prefrontal cortex is trying to decide, you say, oh, animals shouldn't be able to, you know, make the choice. So you should see some kind of loss of function, as like a lesion experiment. Disrupt hippocampus, animal doesn't perform. Yeah, you know, those are pretty typical, but, you know, the fact that animal can't perform it doesn't necessarily tell you that it was because of that specific, you know, it was that specific interaction. It could have been some non-specific disruption. Ideally, what you'd like to have is sort of a double dissociation, that you show that you don't get general loss of function, you get loss of specific function, and it's associated with the sort of the specific independent variable that you're manipulating. In this case, it would be like phase. So you're manipulating the phase, you're measuring behavior, and you don't show that, oh, if you do something at a particular phase, I can disrupt it. What you'd like to show is that certain phases have certain functions. You can disrupt function A at phase A and function B at phase B. That's what you'd like to be able to show. But that wasn't how this was designed. This was designed, really, the idea was, I just want to show that hippocampal activity, phase-locked hippocampal activity in this task matters. And so that's what Josh did. He went in and he locked, he triggered the disruption of hippocampal activity to location on the maze, right here. But of course, you're going to do this, you have to show that, okay, that this region matters, so you need another location. And so the idea is, okay, I'll do the same manipulation, and I'll do it over here and show that it matters over here and it doesn't matter over here. And so this is what he referred to this as the retrieval segment. just because this is where the animal actually presumably retrieves information, makes a choice. And then he referred to this as the encoding segment. Encoding segment only because in this task, the working memory task, the animal has to remember where it came from in order to make the correct decision. In other words, it has to hold in memory, where did I come from, and then decide, use that to make the correct choice here. So 2 segments, behavioral segments, retrieval and encoding. But now he's going to do this, he's going to disrupt hippocampal activity, but he wants to do this in a physiological way, and activity in the hippocampus physiologically is modulated by the theta oscillation. So you really can't just turn off activity. You want to disrupt it as a function of theta. And if you disrupt it as a function of theta, now you have to make another choice. Well, what phase of theta? And so what he decided to do is, because you don't know, is this the right phase? Is this the right phase? He decided to test both phases. And so now you actually have this 2x2, you have this sort of 2x2 condition matrix where you're going to manipulate activity in this segment and this segment at 2 different phases. So I have 2 phases, let's say the peak and the trough. And then I'm going to presumably find, oh, there's like an optimal phase in this segment that's going to disrupt working memory performance, right? That was sort of the explanation. That was the expectation. A 2x2 matrix, 1 quadrant is going to be the effective one, and that will sort of show that you get, the hippocampal dependency. So what did Josh find? He didn't find any of that. So surprisingly, when he actually inactivated hippocampal activity in these, in this region, at particular phases in the retrieval segment, at a particular phase here, in this case the trough, what he found was they got better. Turned off the hippocampus, particular phase, and they got better. You think, well, that's kind of weird. But it wasn't just that segment. He also found that this encoding segment, the segment that was not supposed to be, okay, the prefrontal cortex is not really listening to the hippocampus here, right? He found that he also got an improvement in performance when he disrupted hippocampal activity in the encoding segment, but at the opposite phase. So disrupting hippocampal activity at the peak in the encoding segment enhanced performance. Disrupting it at the trough in the retrieval segment enhanced performance. So not only did he not get disruption of behavior, he got a selective enhancement, a phase by segment interaction. So that's really weird. Why would you get an enhancement of performance? And why would it be phase and segment dependent? So this is where you have to start thinking about, it's this idea that different phases of the hippocampal theta oscillation both encode different information, which I showed you, but interestingly, they also receive different information. So what you see up here in the upper left, this is the hippocampal circuit. We're making all these measurements here in area CA1, which is sort of on the output side of what's referred to as the classic trisynaptic loop. The entorhinal cortex here serves as the kind of the interface to the hippocampus. It provides the primary input to the hippocampus, and there are 2 ways in which information can get to CA1 from the entorhinal cortex. One, there's a direct projection from layer 3 directly into CA1. It's sometimes referred to as the temporal mnemonic path. This is the sort of the temporal, the temporal entorhinal input going into Ammon's horn. CA stands for cornu amanus, Ammon's horn. So this is temporal lobe directly into Ammon's horn, the temporal mnemonic path into CA1. But then there's an indirect path, sometimes referred to as the classic trisynaptic loop, that comes from layer 2 of the entorhinal cortex into the dentate gyrus into CA3, CA3 into CA1. Now, there's also a path that bypasses the dentate, layer 2 to CA3. But when you're in CA1, you have 2 primary sources of input, the stuff coming from entorhinal cortex and the stuff coming from CA3. And CA3 getting slightly different input, layer 2 input, and it's also getting dentate input. So you got these 2 inputs from CA3 and entorhinal cortex. And when you look at both of those are theta modulated, but interestingly, they have a shifted phase preference, and that is that the entorhinal input and the CA3 input coming 180 degrees out of phase. So even though they're theta modulated, like the different phases, like the peak and the trough, are actually receiving information from two different circuits. You got the CA3 circuit and the entorhinal circuit. Now you think, what the heck is the entorhinal circuit doing? What is the entorhinal circuit? So the entorhinal circuit, this is where in addition to the Nobel Prize given to John O'Keefe, Edvard Maybrit Moser also received the prize for the discovery of the spatial encoding in the entorhinal cortex, the discovery of so-called grid cells. And so what they found is that cells, primarily in the superficial layers of entorhinal cortex, in particular regions of the entorhinal cortex, have a grid-like receptive field pattern, suggesting that they're encoding metric spatial information, that they provide that metric spatial information to the hippocampus make place cells. That's how we form our maps of space. At least that was the idea. But what it does suggest is that the entorhinal cortex, which receives convergent multimodal information from across the brain, is sort of taking in all this information. It's taking in visual, auditory, even olfactory information. It's bringing it all together, and it's sort of telling you, okay, this is where you are, right? This is it. This is the current state of the world. Here's current state of the world, hippocampus. And so this direct pathway you can think of as communicating high-dimensional present state information. But what about this pathway through CA3? So the thing that distinguishes CA3, well, even in the dentate shows this kind of sparse sampling, high-dimensional sampling of different environments. One computational argument that's been made is that this sort of high-dimensional representation, there are like 10 times as many dentate granule cells as there are CA3 or CA1 pyramidal cells. And the idea is that this high-dimensional representation allows you to orthogonalize different environments. So it's the way of kind of separating out overlapping patterns. Things that are similar in the real world can be separated and distinguished in the dentate. And then you take that dentate representation, you pass into the CA3. CA3 has this strong sort of recurrent connectivity. It's estimated that like every CA3 cell is connected to like 5% of other CA3 cells. Very dense autoassociative memory, that's the idea. And this dense autoassociative memory, there are different functions you can perform. It basically means that the state of firing of one cell is dependent not only on the input coming from the entorhinal cortex, but on the state of firing of other cells. And that the state of firing of other cells could reflect, let's say, different components of a particular representation, or potentially it could represent different components of like a prior state representation. So recurrent networks have been used and can be used to kind of capture temporal dynamics. You can do temporal coding, using recurrent networks. And so the idea is that, well, perhaps CA3, using mechanisms of autoassociative, sort of memory pattern completion, could either be doing like static pattern completion or potentially like temporal predictive coding. So it's not just, it's using present state to predict like future state. That would be one idea. So you could argue, okay, so I have like present state information coming in directly from entorhinal cortex here, and then maybe I have like this sort of predictive model-based estimation of future state coming in from CA3, and then both of these things are coming in at different phases. So present state, future state, peak, trough. And so now we're going to kind of map that. That's the idea here. So you can map that now onto the task. So you can think the task also has those sort of two components. The encoding segment you can think of as being dependent on present state encoding. I don't need to know where I'm going to be in the next 100 milliseconds or next 500 milliseconds or next second. That's pretty much fixed, but I do need to know where I am right now. So there's a strong dependency on present state encoding here in the encoding segment. Conversely, when I'm up here in the retrieval segment, present state information is of limited utility. Am I in the central segment? Okay, yeah, there's only one location that I can be at. So, this is it. This is the central segment. What I really need to know is, where am I going to be next? And so in the retrieval segment, there's greater utility to the predictive information. Present state useful in the encoding segment, future state useful in the retrieval segment. And each of those, present and future state, can be segregated by phase. And so the argument is that by manipulating here, by disrupting the irrelevant contribution of the other phase, you enhance the relative contribution of the remaining phase. So by disrupting the encoding signal in the retrieval segment, you focus information on the retrieval information, predictive state. Similarly, in the encoding segment, by disrupting the future state information, which is not relevant, you kind of magnify or amplify or you sort of focus the information on the present state information. And so that's the idea that the hippocampus is always capturing present state and future state. But depending on the task, sometimes present state is not as useful. Sometimes future state is not as useful. So this is kind of an artificial manipulation that kind of artificially leverages that, suppressing the irrelevant state information based on task phase. But the suggestion is that this demonstrates, well, it does demonstrate that phase does matter. That for whatever reason, the different phases actually have different functions. And further, it's not that there's a good phase and a bad phase, it's that each phase carries different and to some extent complementary computational functions. And that is, there's not just a good phase and a bad phase, it's that there are different phases that have different utilities in sort of different phases of a behavioral task. And that if I now take that idea, different phases, different functions, different circuits providing, let's say, present state and then future state information, and then map that back onto what I had described here, and that is the encoding of sequential information in the hippocampus as a function of theta phase, now you can kind of see how it makes sense, and that is that there actually is a present state code. So there's the code, for instance, here, that carries very little. This is this sort of early phase information, early theta phase, limited temporal information, high firing rate, a lot of present state information. You say, that's the encoding phase domain. And so that's where I'm getting entorhinal input, that's where I'm reflecting present state, and that's where present state information actually gets used in a behavioral task in a circuit involving hippocampus and prefrontal cortex. The later phase, this region up here, well, I can't get it, well, later phase, up there, that later phase where you have this kind of predictive state, it correlates with future state, firing rate is lower, the correlation of position and time is higher, and that seems to, the expression of that predictive sequential state kind of overlaps with the with CA3 input. So it's the idea that now you have a different circuit, CA3, which is providing future state information, that then correlates with CA1 expressing future state information. Those phases are the ones that the prefrontal cortex would prefer to use when it's actually making decisions. And so this is where, if you look in the hippocampus, and you compare the phase preference in the hippocampus and the prefrontal cortex, you see something kind of interesting. One, that the hippocampus, there's sort of an overall distribution of preferred phase. The certain cells, they just sort of prefer a particular phase. In the prefrontal cortex, there are different cells that have preferred phases, but different cells can prefer different cells have different phase preferences. And so if you're sitting in the prefrontal cortex, the idea is that you have different populations. They're each tuned into different phase channels. Some are tuned into the present state hippocampal channel, some are tuned into the future state hippocampal channel. And so you can simultaneously have the prefrontal cortex kind of listening in, getting from the hippocampus every theta cycle. They're getting this present state, future state. Different populations can be used to, sort of direct different information. So it's sort of the translation of this phase preference in the prefrontal cortex is the translation of this temporally or phase encoded information into sort of distinct distributed populations in the prefrontal cortex, different populations reflecting different phase coded information in the hippocampus. And so I'm going to kind of switch from that. You say everything up to this point has tried to argue that there are biophysical mechanisms that can be used to encode information within the theta cycle related to spatial state information, past, present, future state, that causal manipulations of activity during those states demonstrates their behavioral relevance, and then simultaneous measurements in different areas shows that you get this kind of transient coupling that seems to be correlated with accurate choice behavior, all suggesting, yeah, there's coding as a function of phase, and phase matters. But it's one thing to say that phase matters, and it's another thing to say that that phase information, the sequence information that may be expressed during behavior, in some way is reflected in the memory processing that goes on in the hippocampus. And that is, does any of that temporally structured information persist beyond the behavior itself? And one way to look at that is to look at activity in the hippocampus independent of the behavior. And the two conditions that are nominally independent of behavior, sleep clearly is independent because the animal's in a different context, is not engaged in any behavioral activity at all, and it's in a state, it's in an offline state with diminished arousal where it can't behave. So it's like it completely dissociates any behavioral dependence from any kind of cortical or hippocampal activity. Anything you see during sleep has to be the result of memory, some sort of residual trace of experience. It can't be the result of actual experience because there's no actual experience. To a lesser degree, you can look at this during quiet wakefulness, and that is the periods of immobility where the animal is in an environment, but it's not actively moving. And so we'll compare and contrast those two conditions. One where you're out of the context and you're not behaving, one where you're in the context and not behaving, and you'll see that there are some subtle differences between them, but there are also striking similarities. The striking similarities are that in both cases, the hippocampus of the animal retrieves and reactivates extended sequences, spatial sequences, navigational sequences, that take a very particular form, and that is that they're composed of short chunks lasting about 100 milliseconds, which should remind you of like these theta sequences, and they're expressed during these sharp wave ripple events, and that these sharp wave ripple events can come in like multiple cycles, which again should remind you of the theta oscillations, kind of like offline theta sharp wave ripples, where you get sequences, the expression of remote sequences. And so both in quiet wakefulness and during sleep, sharp wave ripple burst mediated extended sequential replay. The difference between quiet wakefulness and sleep seems to be in the bias in content. During quiet wakefulness, these sequences seem to correspond or seem to be biased more heavily to the present context. You see reactivation of sequences that reflect what the animal has done or what it will do, present context. During sleep, you see much more diverse content, not necessarily because the animal is not in any present context. You see it reflecting, let's say, or sort of potentially combining or integrating multiple prior contexts, right? Memories associated with multiple prior contexts. So it's just really in the bias in content. There's a present state context bias to reactivation in the quiet wakeful state. There's a much more diverse, content in the non-REM or sleep state. You can argue that having more diverse content affords opportunities to kind of explore more compositional relationships between content across different contexts. So you can kind of generalize across contexts because you can combine, evaluate and potentially like synthesize, information during sleep that perhaps you wouldn't do during quiet wakefulness, where you may be using the same capacity for sequential activation to do more directed problem solving. You're kind of evaluating, perhaps planning. So there could be different functions that are subserved by the same basic reactivation property of the hippocampus during sleep and quiet wakefulness. And so the simple kind of the first demonstration is reactivation of sequential information during sleep, just kind of illustrated here. This was some work by Albert Lee when he was a graduate student in the lab way back. Animal sleeps, runs on a little linear track, kind of activating place cells in a sequence, and then goes to sleep again. And you kind of look at activity. This shows some actual, this is actual sleep structure. Red is REM sleep, as I mentioned, paradoxical sleep, with lots of theta. In gray, there's the non-REM. It's referred to as slow wave sleep because of the presence of these slow oscillations in the sort of the 1 hertz range. And it's within these slow oscillations that you tend to see these sharp wave ripple events in the hippocampus. And then this just indicates comparing slow wave activity before running and after running. That's what Albert did. And what he found was, so if you just look at the simple task, recording from, in this case, like 8 place cells, and then these place cells will have place fields along the track. He just orders the place fields, orders the cells based on their place field. So the animal runs to left or right, and you're going to activate these cells in order 1 to 8. When the animal went to sleep after running on this, Albert found that you found the same population of cells, cells 1 to 8, firing in an order that reflected that behavioral sequence, firing sort of 1 to 8. In this particular paper, he did kind of an explicit combinatorial analysis to demonstrate precisely how improbable it would be to get these sequences expressed by chance. And so this was kind of nice because it's a sort of a closed form solution to the reactivation question. You can ask very precisely exactly, what is the fidelity of this reactivation that you're seeing here? Is this a result of just some sort of statistical bias in like ordered reactivation, or is this the infrequent expression of high fidelity extended sequences? And the answer is the latter. That what this represents is high fidelity expression of explicit extended sequences. So it's not simply that there's, that it's like if you take a network, if you took a recurrent network and you kind of ran it, in space, and then you just let the network just go, what you would find is there would be a tendency for, certain like spike order sequences to be expressed just because there's like asymmetry in the like the connectivity matrix. If you had like a sort of a directed learning rule, Hebbian-like rule, it would tend to favor kind of certain cell order sequences. You might find pairwise biases. That's what you'd expect. Just if you just had any kind of associative network with any kind of asymmetric Hebbian-like learning rule, you're going to get stuff, occasionally you're going to get stuff like that. So the question is, are these sequences the product of that simple asymmetric, learning rule, or is there some other mechanism that says, no, when we express sequences, they're going to be high temporal order sequences, long sequences, and that's what Albert found. And that the expression of these little sequences co-occurs with these sharp wave ripple events. So when you see spiking sequence in the hippocampus, it means that the hippocampus is expressing its sort of changed state and is now producing these sharp wave ripple events. And I'm just going to kind of point out here that, when I talk about these sharp wave ripple events, the sharp wave ripple, the sort of the time constant associated with sharp wave ripple is basically the same as the time constant associated with a theta cycle. Same time scale. And in fact, you can use the same simple mechanistic model. In this case, the simple model that I argued, this biophysical excitatory inhibitory model that I used to argue for sequential encoding during behavior, you can use exactly the same model to sort of argue for sequential reactivation during sleep. And the idea here is, so now I'm in the hippocampus, I present to the hippocampus some fixed state, entorhinal cortex produces produces a state, delivers that input into the hippocampus, and then the hippocampus sweeps inhibition from high to low, and as it sweeps inhibition from high to low, it's going to unmask the firing, patterns of these, of the cells receiving this input based on the magnitude of the inputs. So cells that are getting strong input are going to fire first, weaker input are going to fire last. And of course, that static input is simply, it's just the excitatory lines. That's sort of the weighted synaptic input. So if during behavior, synapses capture the magnitude of this, right? of these inputs, when I re-express those inputs, re-expressing the magnitudes of those of the inputs, and then I sweep inhibition, adding in sort of the temporal dynamics, I simply recover the time sequence. So same mechanism, sweep inhibition to encode sequences and to recover or replace sequences. Same mechanism. Now it just happens that this dynamic modulation of excitability, in particular of inhibition in the hippocampus, arises as the result of projections from the primary cholinergic, forebrain cholinergic nucleus, the medial septum. Medial septum provides modulation of cholinergic input at the hippocampus. Cholinergic tone is sort of arguably necessary and sufficient, but is highly correlated with the expression of the theta or the non-theta state. High cholinergic tone, get theta oscillations. Low cholinergic tones, low cholinergic tone, you get sharp wave ripples. You know, high cholinergic tones is associated with active locomotion, also REM sleep. Low cholinergic tone associated with non-REM sleep, quiet wakefulness. So you can fiddle with it. You can go in there, you can kind of tweak this, dump in, take a slice, dump in acetylcholine, you can get theta. So there's a good correlation there. But then there's another population of medial septal cells, non-cholinergic, they're GABAergic cells. And they project along with the cholinergic cells. It's just that the cholinergic cells come in, they kind of dump acetylcholine, primarily, you know, in sort of dendritic regions of the, you know, pyramidal cells. The GABAergic cells come in and they terminate on inhibitory cells. And it's the GABAergic cells that are the ones responsible for pacing two things. One, theta oscillation. Two, kind of sharp wave ripple. So what happens is, medial septum, for theta, you just, you know, you get this kind of dynamic, you know, rhythmic modulation of inhibition. For sharp wave ripples, you get this transient disinhibition. Medial septum says, okay, we're gonna, you know, drop acetylcholine, and then I'm gonna transiently drop inhibition, unmasking whatever the inputs are at that point. I'm gonna unmask them in the order in which they occurred, thus retrieving the spatial sequence information that was already captured, it was already present in the inputs. So the idea is that the inputs, this kind of spatial magnitude, and that is that there's already kind of a magnitude position relationship. There are gradients in the world. If the inputs already have a natural gradient structure, I can translate that natural gradient structure into time sequence information by simply doing this. And so, in a sense, it's not like creating temporal sequences, it's sort of extracting the inherent sequential structure from the inputs that are present in the world, given the natural constraints on the way in which I interact with it. So it's sort of the ecological, it's just kind of extracting the stuff that's already there rather than creating an independent temporal code, capturing the temporal code that's already there. Yeah. So acetylcholine, one thing is it's relatively slow, and the mechanism by which acetylcholine acts is sort of on a longer time scale. It's not on a single theta cycle time scale. So acetylcholine is really used to kind of regulate the transition between the different states. So when I showed you like that illustration, like there was theta and then there was sharp wave ripples, I said, yeah, you know, it takes about half a second, the transition. That's sort of the time scale of cholinergic modulation. It's kind of switched from the active locomotion quiet wakefulness. It's on the time scale of, you know, like hundreds of milliseconds to, you know, to seconds. This, the GABAergic modulation, that's on the time scale of milliseconds to tens of milliseconds. So that's inhibition regulating the rapid dynamics. You know, these neuromodulators regulating the slow transition between the different states. So when you record those inhibitory cells, yes, that's exactly what they do. They exactly pace the theta rhythm. In fact, you can use those as sort of a good proxy for the theta oscillation. So they fire at high rate. That's the thing about the inhibitory cells. They fire at high rate, which gives them sort of high temporal fidelity. They can sort of capture rapid changes in, you know, excitation inhibition. And so that's what you get: high firing rate, theta phase locked dynamic, you know, regulation. So that's what inhibitory cells actually look like. I mean, so this is a cartoon that's, again, sort of inferring this is what inhibition onto those cells look like, given that this is actually what inhibitory firing rate looks like. You know, another thing, you know, we're almost done here, but again, just for the biophysics, you know, I love the biophysics because the biophysics, you can think it's like a cartoon, but it explains a lot. In principle, it explains everything, right? If we knew the accurate biophysics, we would know the answer to how the brain works. So when you look at the details, so this is a cartoon. I remember when we first published this, people criticized, that can't be right, because they get this linear thing, but you said the inputs are theta modulated too, right? And it's like, yeah, you're right. I mean, the input excitation is going to be theta modulated. It's true. So you have to, this is really like the envelope. There's an envelope you actually, so if I really drew this, right, I would have to draw like the theta, this would be theta modulated, right? But now, let's suppose I do that just the way I drew it, right? Where the theta, the excitation is also zero phase locked with inhibition. And now I ask, okay, where does this work? Where does excitation exceed inhibition? If it's zero phase locked, this thing doesn't work, right? The only way it works is I got to phase shift it. Now, if I phase shift this by 90 degrees, right? So that now it's like this, right? Then it works perfectly. So of course, when you actually look at the phase relationship between the excitatory cells and the inhibitory cells, they're 90 degrees phase shifted, because that's the only way this would work. Now you can say, well, they don't have to be, because there are actually a lot of inhibitory cells. I just shown one here. Inhibitory cells are very diverse. Different inhibitory cells firing at different phases. So it's not in any way necessary for excitatory cells to be 90 degrees phase shift. They could be perfectly phase locked with the primary cells here. These would be the local parvalbumin positive inhibitory cells that regulate inhibition that governs firing. Okay, that's the thing. There are other inhibitory cells. They target different parts of the dendrite. They target other inhibitory cells. They fire at all kinds of different phases. So you can have pyramidal cells firing at any damn phase you wanted, if you really wanted to. It just, the phase that you actually get is the 90-degree phase that would be required for this simple biophysical mechanism to work. Which to me, again, it's not definitive. It just says, if you could choose and you chose that, there probably is a reason you chose that. Why do you have them shift in 90 degrees out of phase? And that's because you need to shift them 90 degrees out of phase if relative excitatory and inhibitory level matters. Because that's the only way you can make it matter. That's the only way relative excitation inhibition can be evaluated, is if they're shifted 90 degrees out of phase. And this is just one argument that one way they would matter is in the timing of action potentials. So we're basically done, even though I didn't get to the memory reactivation. So you get like sequence reactivation. I kind of went through some of this before, talking about you get reactivation of these hippocampal sequences. You also see them in the visual cortex. You can manipulate these things during sleep. You can bias the reactivation by biasing cortical activity. And then I just wanted to point to this is all published stuff, so I think the one thing I wanted to point out, this is the kind of the extended replay. I mentioned the reverse time order, but the thing I really wanted to point to is this. It is on a longer track. When you look on a longer track where you have longer sequences, and then you look at when are those sequences expressed, you find during quiet wakefulness, you have these periods lasting on the order of about 500 milliseconds, which is exactly the time scale of what referred to as the slow oscillation during slow wave sleep. And it's during those periods which are governed by sharp transitions in relative excitability, so-called on and off periods. Periods where like all the cells are silent and then suddenly there's lots of activity and then they're silent again. During these kind of elevated up periods, what you see in the hippocampus is when you decode activity over 500 milliseconds, you see these extended sequences. The extended sequences are actually composed of multiple adjacent sharp wave ripple events. So you get these bursts of sequential sharp wave ripple activity that are somehow chained together. And an interesting question is how do you chain them together? And what does that actually represent? So within the sharp wave ripple, the argument is that the sharp wave ripple represents, just as the theta sequence represents, it represents sort of the high confidence spatial correlations or predictive spatial contingencies. If I know where I am, like right now, with very high confidence, I know exactly where I'm going to be in the next couple hundred milliseconds. After that, it gets sketchier. And so you can kind of think of there being, if you're sort of constructing a graph each of these sharp wave ripple events represents a node in the graph, where the node represents a state, where the state in this case is not just a static state, it's a temporal sequence state, but it's a temporal sequence state in which the coherence of the, or sort of correlation of the locations is relatively high. And that during these periods of quiet wakefulness or sleep, you chain these things together in a way that potentially reflects some sort of active search of this graph where you're trying to evaluate future state outcomes. And consistent with that idea that the expression of extended sequential state is actually a search of some sort of like state space contingency graph where there's like a, there's a result at the bottom, right? It's not just like an organized series of states. It's like you start from one point and you're trying to evaluate where you end up, and then you're going to choose different paths based on that outcome, right? Some optimization based on the sort of the terminal state of a search of a graph. That comes from stuff that, again, is published, and I won't show you the details, but when we record activity in areas that like the VTA, that reflect dopaminergic signaling that correlate with reinforcement or reward outcome, and so they reflect, again, sort of the results of some sort of behavioral contingency. What you find is reward signals tend to occur or expressed here at the end of these extended sequences, and reward signals, when they're expressed, reflect in the hippocampus the expression of like goal states. So hippocampus replays sequences. If the sequence ends in a goal, reward information associated with that goal is also expressed. Again, suggesting there's more than just like some sort of passive expression of like random elements, and more a directed search through some sort of behavioral state contingency graph for the purpose of sort of optimizing behavioral choice. Now, one caveat in that is that's what you see during quiet wakefulness, where I argue quiet wakefulness and non-REM sleep are very similar. When you look during sleep and you look at the same activity, you see these same events, same upstate associated activity, but you don't get the reward contingency expressed at the end, suggesting that, again, by just sort of pairing this, you can have like this deep graph search, relatively deep, search of this like state space graph. In one case, if you're trying to optimize for performance, you throw a reward in there and say, okay, what's the result of this? But during sleep, you're not actually looking at reward, you're trying to look for something else. And the idea that during sleep, you have a lot more sort of heterogeneity or diversity of content suggests that you might be using these events in some way to generalize, to synthesize, same capacity, minus reward, to perform some other kind of mnemonic processing. And I also just point out that that's not because reward is not relevant during sleep. You can actually demonstrate that if you pair reward signaling with these events, you can reinforce behavior. You can even reinforce behavior that has never been expressed during active wakefulness, showing that you can learn stuff in sleep that you never even experienced during wakefulness. But for some reason, the brain seeks to avoid that during normal sleep. And so I think that's sort of a functional question that has yet to be answered. So with that, I'll just end it.